The overall goal of this protocol is to overexpress and purify codon-optimized human cis-prenyltransferase under non-denaturing conditions and to assay its enzymatic activity. The procedure is demonstrated with the eukaryotic long-chain cis-prenyltransferase dihydrodolycyl diphosphate synthase, or DHDDS.
This method can advance our understanding of isoprenoid synthesis and provide key insights into the pathophysiological mechanism of retinitis pigmentosa related to mutations in DHDDS.
The main advantages of this technique is that it's simple, cost-effective, and time-saving. It also can be generalized for high-quantity production of other cis-prenyltransferase proteins.
Begin this procedure with cloning and expression of human DHDDS as described in the text protocol. Resuspend the cells in buffer A, 1 microgram per milliliter of DNase-1 and a protease inhibitor mixture. Then, homogenize the resuspended cells using a glass Teflon homogenizer. Disrupt the cells with a microfluidizer or equivalent at 12,000 to 15,000 psi. Next, centrifuge the cell lysate at 40,000 times G for 45 minutes at 4 degrees Celsius. Recover the supernatant containing the soluble fraction. Load the supernatant onto a 5 to 10 milliliter cobalt immobilized metal affinity chromatography column with 10 millimolar imidazole. After loading into a fast protein liquid chromatography machine, perform a thorough washing of the column with buffer A and 10 millimolar imidazole in order to reduce